Hello, this is Akram Jafar, and today I'm going to deal with the renal system, concentrating mainly on the kidney. The functional unit of the kidney is called the nephron, and there are about 1 million of them. Part of the nephron is present in the cortex, and part which is present in the pyramidal-shaped medulla. Each nephron is composed of a corpuscle, renal corpuscle, and a tubule. Some of these corpuscles are located close to the medulla, but in the cortex, so they are called juxtamedullary nephrons, and the others are located away from the medulla, so they are called cortical nephrons, and these juxtamedullary nephrons, they have more ability to concentrate the urine than the cortical nephrons, but both of them, they are located in the cortex. Juxta means close to. Now, in the renal corpuscle, it is the site of blood plasma filtration. So the corpuscle consists of a tuft of capillaries, which is called the glomerulus, to increase the surface area of filtration. Then there is a Bowman's capsule or a glomerular capsule, a double-walled cup. One pole of the cup is called the vascular pole. It's through which the blood vessels enter and leave. So we have an afferent arteriole and an efferent arteriole. And the other side of the cup is called the renal pole, where it is continuous with the renal tubule, the proximal convoluted tubule. So this is the proximal convoluted tubule here. It is convoluted, folded on itself. It leads into a U-shaped loop, which is called the loop of Henle, has a descending limb and an ascending limb. Part of the limbs is thin part and the other part is thick part. The thick part is lined with cuboidal epithelium and the thin part is lined with simple squamous epithelium, and it leads into a distal convoluted tubule, another tubule which is shorter than the proximal convoluted tubule, that leads into collecting ducts, and then papillary ducts become larger and larger and open at the tip of the renal papilla, and so droplets of urine are going to be taken into this small cup, which is the minor calyx. In order to produce urine, filtration should take place in the renal corpuscle, and what facilitates this is that filtration is driven by pressure. So you can see the afferent arteriole is wider than the efferent arteriole, and this results in a higher pressure of blood in the glomerulus, which will push most of the plasma of the blood to be filtered into the Bowman's capsule. The Bowman's capsule is lined by simple squamous epithelium as a parietal layer, but the visceral part here, which is close to the blood vessels, look at these capillaries which contain RBCs. The Bowman's capsule is lined by cells which are called porocytes. Porocytes mean that they have feet. This is a higher magnification. You can see the blood vessel, the capillaries, lined by endothelial cells, fenestrated endothelial cells. There is a basement membrane, and then these are the porocytes. They have large feet, primary and secondary processes. So large feet and small feet. Together, the porocytes, the basement membrane, and the fenestrated endothelium, together they constitute what we call the filtration membrane. Most of the contents of the blood are filtered except the blood cells and the large molecules, like large protein molecules. So if this filtration membrane is destroyed in diseases of the kidney, then this will result in loss of protein and blood in the urine. Let's go back here. You can see that there are other cells which are called mesangial cells, and these are like supportive cells. This is just a diagram. We will not be able to differentiate these cells in a conventional histological stain. So let's look at a conventional histological stain of the kidney. You can see the renal cortex. Look at these dark areas here, dark spots. These are the glomeruli. Glomeruli are only present in the cortex, while in the medulla you don't expect to find glomeruli. It's only the collecting ducts, papillary ducts, ascending and descending loops of Henle, but there's no proximal or distal convoluted tubules. This is a renal cortex. It shows the renal corpuscle. Look at the tuft of capillaries, and this is the Bowman's space. This is a higher magnification. Look at the parietal layer of the Bowman's space, simple squamous epithelial cells, and this is the glomerulus. A lot of cells are located here, but we will not be able to differentiate these cells. Some of them, the nuclei, belong to the podocytes, some of them they belong to the endothelial cells, some of them belong to the mesangial cells. All the other profiles, surrounding profiles, are of the tubules, proximal and distal convoluted tubules. Both these tubules are lined by cuboidal cells. The cuboidal cell has a centrally located nucleus. One side of the cytoplasm, which is this one, is the luminal surface. So if this is the tubule and these are the cuboidal cells, this is the luminal surface. The luminal surface for the proximal convoluted tubules, because most of the reabsorption takes place in the proximal convoluted tubule, so we need more surface area for reabsorption. Therefore, the plasma membrane is folded into what we call microvilli, small finger-like projections called microvilli to increase the surface area. These microvilli can only be seen as such in the electron microscopy at a very high magnification, but in the light microscopic level, they appear as a fuzzy border here, which is called brush border. And so you can see that in the proximal convoluted tubule, there is a fuzzy and hazy border at the luminal surface. In addition to that, because there is a lot of activity taking place here, a lot of absorption taking place here from the lumen, then there are a lot of organelles in the cell, like transport vesicles. There are a lot of mitochondria that provide energy. And so the cytoplasm in the proximal convoluted tubule stains darker. So let's go back here. You can see that some of these, especially here and here, most of them actually, the profiles, they are darker stained if they are compared with this. For example, the lighter stained are the distal convoluted tubule. They don't have a brush border, but you can see that in the proximal convoluted tubule, they have this brush border. The cytoplasm is darker in color. And in addition to that, we can see more profiles of the proximal convoluted tubule than the distal convoluted tubule, and the reason is that the proximal convoluted tubule is longer than distal convoluted tubules. A point should be mentioned about what we call the juxtaglomerular apparatus, and this is where an afferent arteriole makes contact with a distal convoluted tubule. The juxtaglomerular apparatus consists of cells which are called macula densa cells. This is the thickened part of the distal convoluted tubule. You can see that the cells are more columnar, they are no more cuboidal, and their nuclei are close to the lumen. It's called macula densa, and these cells in fact are chemoreceptors. They sense the concentration of the urine which is present inside the lumen. And then the other types of cells are modified smooth muscle fibers which are present in the wall of the arteriole, and these are called the juxtaglomerular cells. In fact, they are baroreceptors. They sense the blood pressure in the arteriole. And depending on these sensory information, they secrete a hormone which is called renin that regulates the blood pressure. This is in the medulla. You can see that there are no glomeruli. It's only tubules. And these tubules, they are not proximal, not distal. They are the ascending and descending loops of Henle, as well as the blood vessels that accompany them, which are straight blood vessels, not like the glomeruli. These are called the vasa recta. Some of the profiles, they have cuboidal epithelium, so they represent the thick part of the loop of Henle or collecting duct, and some of them are lined with simple squamous epithelium, and they represent the thin part of the loop of Henle. This is at the tip of the pyramid. This is the renal papilla. You can see that they are all collecting duct, and the space here is the space that belongs to the minor calyx. The minor calyx is lined by a transitional epithelium. This is the epithelium that lines the excretory passages of the renal system. You can see here, this is a section of the ureter. The lumen has a mucosa which has a epithelium which is transitional epithelium. The transitional epithelium is a stratified epithelium. It has multiple layers, but it's called transitional because the cells, especially the cells at the surface layer, like these ones, they can change their shape depending on the condition of the organ. You can see here, this is when the organ is empty. You have these dome-shaped cells, and then the cells become flattened when the organ becomes distended. So the epithelium changes its shape, and that's why it's called transitional epithelium. And because it is exclusive to the urinary passages, then the other name of the epithelium is called urothelium. Back to the ureter, you can see that the ureter has smooth muscle in its wall, and these are important for peristalsis to push the urine from the kidney to the urinary bladder. And this is the urinary bladder. Again, it is lined by transitional epithelium, but there are a lot of muscle fibers in the wall, and this will help in the mass emptying of the urinary bladder when they contract during urination. Note that in the urinary bladder, because it is lined with transitional epithelium, which is a stratified epithelium with tightly bound cells, then there is no concentration of urine taking place. The urinary bladder only stores urine, it doesn't concentrate urine. This is how the urinary bladder differs from the other bladder, which is the gallbladder. And the gallbladder stores and concentrates bile, but the urinary bladder only stores urine. No concentration of urine takes place in the urinary bladder.